We see warning labels on pretty much everything, right? From cigarettes to cleaning supplies. But have you ever just stopped and wondered, how? How can a chemical actually cause something like cancer? Well, it's a journey, and it starts with a single, invisible spark inside just one of our cells. Today, we're going to follow that journey step by step to really get the science behind this whole process. So, let's dive in.
Yeah, we've all heard it. We've seen the headlines. This causes cancer, that causes cancer. But what is really going on inside our bodies when we're exposed to something like tobacco smoke? How does a substance from the outside world manage to hijack our own cells and, turn them against us? That's the big mystery we're going to unravel today.
And this isn't just some abstract scientific question, it's a really critical public health issue. According to the World Health Organization, cancer is responsible for nearly 1 in every 6 deaths around the world. I mean, that is a staggering number. And understanding the root causes is without a doubt our best weapon in the fight to prevent it.
Okay, so before we can get into how chemicals attack our cells, we need to make sure we're all on the same page. Let's start with a quick primer. What exactly is cancer?
First thing you need to know is that cancer isn't just one single disease. It's actually a super broad term for a whole group of diseases. But they all share 2 key things. First, certain cells basically forget how to stop growing. They just divide and divide uncontrollably. And second, they don't respect boundaries. They can invade surrounding tissues, and that's what makes them so dangerous.
This visual right here is a perfect comparison. So on the left, you've got a benign tumor. Think of it like a crowd that's staying put behind a velvet rope. The cells are multiplying, sure, but they're contained. A malignant tumor, on the right, well, that's when the crowd breaks right through the rope. These are the cancerous cells. They're invading nearby tissue, and they've got the potential to spread.
So if cancer is the outcome, what's causing it to start in the first place? All right, let's meet the main culprits of our story.
The official name for these cancer-causing agents is carcinogen. You can think of them as these tiny little saboteurs. Their main weapon? They get inside our cells and they mess with the instruction manual, our DNA. And by doing that, they can introduce errors, what we call mutations, that can set a cell on a really dangerous path.
And these aren't just some rare chemicals you'd only find in a high-tech lab. Carcinogens are actually surprisingly common in our day-to-day lives. We're talking about things in tobacco smoke, the char on your grilled food, alcohol, even diesel exhaust. They've really become a part of our modern environment, for better or worse.
Now, scientists usually lump carcinogens into 3 main buckets. First, you've got physical ones, like the UV radiation from sunlight. Then there are biological carcinogens, which can be viruses, like HPV. But for our purposes today, we're going to focus on that big middle group, the chemical carcinogens, things like asbestos and tobacco smoke. This all raises a pretty big question, right? With so many things out there, how can we be sure that something is truly a carcinogen? I mean, you can't just guess. Turns out there's a really rigorous system for this. The gold standard for this comes from the International Agency for Research on Cancer, or IARC, and they make a really important distinction. Their classification system isn't about how high the risk of cancer is. Instead, it's all about how certain the scientific evidence is that a substance can cause cancer at all. So when something like tobacco is in group 1, it means the evidence is just undeniable. They call it hazard identification, not risk assessment, and that difference is key.
Okay, so now we know what carcinogens are. Let's get to the main event. How do they actually do their dirty work at the molecular level?
So, chemical carcinogens have 2 main playbooks. On one side, you have the genotoxic ones. Think of these as the direct attackers. They get into the cell and physically damage the DNA. Sometimes they form these things called DNA adducts, which are kind of like sticking gum in the gears of your genetic code. Then, on the other side, you've got the non-genotoxic agents. These guys are more subtle. They don't attack the DNA directly. Instead, they create a hostile environment around it, through things like chronic inflammation that makes cancer more likely to grow.
Let's use some real-world examples to make this a little clearer. So in the genotoxic corner, you've got compounds in tobacco smoke that literally bind to DNA, forming those adducts we were just talking about. But in the non-genotoxic corner, you have something like asbestos. It doesn't directly mutate the DNA. Instead, the tiny fibers trigger chronic inflammation in the lungs, which creates this perfect storm where cancer is more likely to pop up. So 2 totally different paths, but they can both lead to the same devastating result.
It's also really important to get that this isn't an instant thing. A single exposure doesn't just immediately lead to a tumor. Chemical carcinogenesis is a long journey that happens in several distinct stages over time. Seriously, from the first moment a carcinogen damages a cell to the point where you can actually detect a tumor, we could be talking years or even decades. It's a slow process, and it unfolds in 3 clear stages. A really great way to think about this is like starting a fire. First, you have initiation. That's the initial spark that hits the kindling. Then comes promotion, which is like fanning those flames, helping that little spark grow into a small, steady fire. And then finally, you have progression. And that's when the fire just roars out of control and starts to spread.
And this diagram here, this shows us that exact process, but in cellular terms. See how it starts with just one single cell getting hit during initiation? Then, in the promotion stage, that damaged cell starts to multiply, making more and more copies of itself. Finally, during progression, the cells get more mutations and transform into that malignant tumor.
So, let's break down each stage. Initiation is that very first hit. A carcinogen causes a permanent, irreversible mutation in a cell's DNA. But, and this is a really critical point, this single cell is not cancerous yet. It's just been initiated, kind of like it's been primed for trouble down the line.
Okay, next up is promotion. This is where other things, and they don't even have to be the original carcinogen, encourage that initiated cell to multiply. It starts building this little clone army of cells that all carry that first mutation. And what's really important here is that this stage can actually be reversible. If you remove the thing that's promoting the growth, sometimes it can all just grind to a halt. But if that promotion keeps going, the cells can enter progression. This is the final stage, and it's irreversible. The cells start piling up even more mutations, becoming more and more unstable and aggressive. This is where they get all the classic hallmarks of cancer, that out-of-control growth and the ability to invade and spread.
So, let's tie this all together. What have we learned? Well, at its core, cancer is a disease of uncontrolled cell growth. It's kicked off by carcinogens that damage our DNA, either right up close or indirectly. And maybe most importantly, this isn't just one single event. It's a slow, three-act play: initiation, promotion, and progression.
And that brings us to one final, really crucial question to think about. Knowing this is a long chain of events, a process that takes years to unfold, and with a potentially reversible stage right there in the middle, where's our best shot to intervene? Is it trying to prevent that very first spark of initiation? Or is it more effective to try and stamp out the flames during that promotion phase? Finding the answer to that, well, that's at the very heart of cancer prevention and research today. Thanks for joining me.